on this computer.
Yeah, well, we're gonna split it up, I think, and have like two groups. So one will go into the lab first, to do like cell culture stuff. And then one of
Okay.
these and the other will go into
Guess it's
cell
recording.
culture.
The screen is being shared,
So we're ready to go.
so we're ready to go.
Okay, I will start.
Yes, sound is up.
Okay, good morning, everyone. It's time to begin. So welcome, everyone. First, you want to welcome Sonia's family and friends who traveled across the country to be with us today. Welcome. So I'm Greg Hermanix. I've been serving as Sonia's major advisor for the past 5 years or so. And so today what we're going to do is during the first hour or so, Sonia will give a talk about her thesis work. And that'll be the public portion of this, and people are encouraged to ask questions, except for the committee, which will reserve their questions. Because right after the public section, we'll have a private, fun exam session with Sonia, where they will ask all their tough questions. So just whoever's online.
Everyone on Zoom, please mute yourself. Thank you.
Okay, so a little bit about Sonia's background for those who don't know her very well. Sonia came to us from UC Santa Cruz, and she got a degree in 2017 in neuroscience. She joined the PIT program, and shortly after that, joined the molecular pharmacology graduate program, rotated in my lab and decided to stay. And so one of the things that impressed me most about Sonia was how she was willing to take on difficult, risky projects with great enthusiasm. And when she first joined the lab, I pitched a couple of far out ideas that were almost doomed to fail from the start, but if they worked, they would be really great. But not surprisingly, those experiments crashed and burned pretty badly. But Sonia kept up her enthusiasm, continued on, took on some other high-risk projects that eventually panned out and formed the basis for the thesis. So Sonia's been quite successful. She has 2 really solid first author publications already. She has another one that's about to be submitted hopefully in a couple of weeks. She also has 3 additional co-authored publications. She has numerous abstracts from various presentations that are local and national meetings. She's been very successful getting funding to help support her time in the lab. She was supported by the Pharmacological Sciences T32 training program for a couple of years. She was awarded the prestigious DeGroat Fellowship in Neuropharmacology. She got a merit award from the Research Society on Alcoholism. And most impressively, she successfully competed for an individual F31 predoctoral research fellowship from the National Institute on Alcohol Abuse and Alcoholism. And lastly, when Sonia's done with her time here in Pittsburgh, she's headed back to California. She has a postdoctoral position at the University of California, San Diego, where she will continue her work in a very prestigious addiction lab of Olivier George. So without further ado, Sonia, please give us your presentation.
Thank you, Greg, for your lovely introduction. And I will jump right in. I'll be presenting today on my dissertation titled Long Non-Coding RNA Dysregulation Alters Ethanol Drinking Behavior and Ethanol-Related Phenotypes. Alcohol use disorder is defined as a chronic and relapsing brain disease that is characterized by the inability to control alcohol intake and despite negative health consequences. According to the National Survey of Drug Use and Health, approximately 85% of adults in the United States report consuming alcohol, and roughly 11% of adults in the United States have been diagnosed with AUD. However, of those who have been diagnosed with AUD, only about 7% actually receive treatment. There is a large global burden that accompanies alcohol use disorder. Alcohol is the first leading risk factor for premature death and disability of people aged between 15 and 49. Alcohol also contributes to over 200 different diseases and injury-related health conditions. There's a large economic burden in the United States. with alcohol costing roughly $250 billion due to alcohol misuse in 2010 alone. In the United States, there are also many deaths attributed to alcohol misuse, with about 95,000 people dying annually from alcohol-related causes, which does make alcohol-related deaths the third leading preventable cause of death in the United States. It is very well known that alcohol can impact the non-coding RNA transcriptome, which leads to modulation of non-coding RNA pathways. This is a schematic summary of alcohol-mediated regulation of non-coding RNAs, with the yellow boxes representing non-coding RNA regulatory roles at both the DNA and the RNA level. It is, however, not well understood how these cellular changes underlie the larger picture of alcohol addiction. So transcriptomic changes are thought to underlie the transition from recreational drinking to problem drinking, and persistent transcriptomic changes may contribute to relapse over time. Non-coding RNA dysregulation has been implicated in AUD already. However, this is still a relatively novel field. Additional research is still needed to determine whether newly discovered transcriptional changes can be mechanistically linked to ethanol drinking behavior. Long non-coding RNAs specifically are emerging as key regulators of the transcriptome. They are a subclass of RNAs that exceed 200 nucleotides that do not encode a protein, and they are located throughout the genome. Long non-coding RNAs are known for their roles in epigenetic regulation. They can act as guides, scaffolds, and decoys to help maintain cellular homeostasis. Their expression is very tightly regulated. They can be tissue or cell type specific and vary spatially, temporally, or in response to stimuli such as alcohol. There are thousands of long non-coding RNA genes and even more transcripts, and many of those do show brain-specific or brain region-specific expression. However, out of these thousands of genes, only a handful have been characterized for their roles in activity and regulation. Long non-coding RNAs also hold high therapeutic promise. The tight level of specificity that they are under offers them up as superior targets for proteins for disease pharmacology. So now, knowing that alcohol can impact the non-coding RNA transcriptome and that long non-coding RNAs are regulators of the transcriptome, next question is, can individual long non-coding RNAs modulate drinking behavior? So that brings me to my central hypothesis that ethanol-responsive long non-coding RNAs are critical hubs of molecular networks that can act as determinants of ethanol consumption and ethanol-related behaviors. So the research completed herein addresses a gap in knowledge surrounding AUD mechanisms. Ethanol can dramatically alter the transcriptome, but it is still unknown how this dysregulation shifts the propensity to consume ethanol and alter behavioral responses to ethanol. So that brings me to my specific aims. My first aim is to test the hypothesis that long non-coding RNA TX2 contributes to ethanol drinking behavior and responses to ethanol. Aim 2 is that mutation of ethanol responsive long non-coding RNAs identified from hippocampal competing endogenous RNA networks are sufficient to decrease ethanol drinking and ethanol related behaviors. And my third aim is to test the hypothesis that mutation of the long non-coding RNA Gas5 promoter in the medial prefrontal cortex will increase ethanol drinking and ethanol related behaviors. 3 CRISPR-Cas9 techniques were applied. CRISPR-Cas9 functions via a complementary cRNA that binds to the genomic location of interest, as well as to an invariant tracer RNA. And these two together form the guide RNA, which then recruit Cas9 to the desired genomic location, and cleavage occurs 3 base pairs upstream of the PAM sequence. So the 3 techniques that I used for aim 1, I used a traditional CRISPR-Cas9 global genome editing technique via embryo electroporation. Aim 2 uses an accelerated CRISPR-Cas9 global genome editing technique via embryo electroporation. And aim 3 utilizes an acute and brain region specific genome editing technique via stereotaxic AAV injection. This is the project workflow that I followed throughout my 3 aims, and I will be referring back to this throughout my presentation. So that brings me to my first aim, that TX2 contributes to ethanol drinking behavior and responses to ethanol. Now, I need to make it very clear that this aim was a collaborative effort across 6 different labs and 2 universities, all within the INEA-N consortium. So all work completed by collaborators is noted next to their respective data. RNA sequencing data from postmortem human brain samples from the cortex, the basolateral amygdala, and the nucleus accumbens identified a human long intragenic non-coding RNA as being selected for functional interrogation, which I will get to on the next slide. This was done by collaborators Dr. Mayfield and Ferris. The murine homologue to this human long non-coding RNA is referred to as GM41261, which from here on out, I will be referring to as TX2. Like its human homologue, TX2 is the natural antisense transcript to the oncostatin M receptor within the first intron. Some long non-coding RNAs have been shown to directly regulate their neighboring genes in a cis manner. So this and previous studies have identified OSMR as being in the middle of a vast AUD-linked network. So this provided a strong foundation for TX2 functional interrogation. A deletion in the putative promoter and first exon was made in order to mutate the gene of interest without altering potential regulatory roles of the intronic region of OSMR. So a 306 base pair deletion was made, and following genome editing, cohorts of TX2 mutant mice were used for behavioral and electrophysiological analysis. And this was done in the Humanics lab with Carolyn Ferguson prior to my joining of the project. So the mutation was made and behavioral responses were measured, and this revealed an interesting ethanol responsive phenotype. Now, I will discuss what these results mean in a moment, but for now, the main takeaway is that the TX2 mutant mice produced a highly interesting and ethanol responsive phenotype. And at this point is when I was brought in on the project to complete the molecular analysis of these mutant mice. This work was all done by collaborator Dr. Blednov at the University of Texas at Austin. So to analyze the mutation at the DNA level, primer sets that flank the deletion site were used. So at the DNA level, you can see a clear wild type band at roughly 500 base pairs. And when the deletion is made, you can see in our TX2 mutants a clear roughly 300 base pair deletion, which is consistent with the 306 base pair deletion that was made. So this was also completed at the RNA level using the same primer sets, as well as a primer set that are downstream of the deletion site. And this is to assess whether the mutation altered transcription of the RNA product. And to our surprise, the fact that the F1R1 primers were able to produce an RNA transcript indicates that the RNA product transcription start site is actually farther upstream than the NCBI gene annotation. And the downstream primers show that the gene is still being expressed and is not knocked out. So we can see here that the deletion is harbored and maintained within exon 1. After extensive RT-PCR and sequencing results, this is the annotation that I have designed for TX2, which incorporates exon 1, intron 1, exon 2, as well as going both farther upstream and downstream. And then exon 3A is now referred to as exon 2. So following RNA analysis, it was of interest to quantify the amount of RNA to determine if this mutation altered TX2 expression levels. And what we can see between our control and TX2 mutant mice is that TX2 is still expressed at normal levels within the cortex. And as I mentioned previously, TX2 is the natural antisense transcript to the oncostatin M receptor. So it was of interest to also measure OSMR RNA levels. And you can see here that the TX2 mutant mice had no difference in Who has me on? There is no significant difference in OSMR expression levels between our TX2 mutant mice and controls. Who has me on Zoom? Okay. So this shows that the mutation has been harbored internally. There is no change in the expression levels. However, there is still a very interesting ethanol responsive phenotype that was produced. So as I mentioned, TX2 was a uncharacterized gene and now incorrectly annotated gene as well. So it was of interest to determine differential gene expression between males and females and across brain regions. So this was done first in the cortex between TX2 male and TX2 females, showing that there is similar levels of expression and there is no significant difference across sex. This was also done in 6 different brain regions: cortex, cerebellum, hippocampus, pons and medulla, hypothalamus, and olfactory bulbs. And there was relative same expression of TX2 across all brain regions with no significant difference when compared to cortex as the control. It was also of interest to analyze the cellular and subcellular localization of TX2. And so this was done via in situ hybridization with or without the coupling to immunohistochemistry. And TX2 expression was visualized in 4 prominent neural subtypes: microglia, astrocytes, neurons, and oligodendrocytes. So to first consider microglia, I know it's a little difficult to see. Turn the lights down. Thank you. So first, showing in microglia, TX2 expression is shown in red, IBA1 positive microglia are shown in green, and DAPI stained nuclei are shown in blue. The merged image is on the right, and when I enlarge this boxed cell, what I hope you can appreciate is clear signs of co-localization between IBA1 and TX2 as shown by these yellow puncta. And what is also clear from this is that TX2 does not appear to localize to the nucleus, but appears to localize to the cytoplasm in IBA1 positive microglia. This was done next in astrocytes with TX2 again shown in red. GFAP positive astrocytes have been shown in green. DAPI stained nuclei are shown in blue. And when I blow up this, enlarge this cell, you can see in this GFAP positive astrocyte, there appears to be no expression of TX2 or co-localization with either the DAPI stained nuclei or the external cell boundaries. So this indicates that TX2 is not expressed in astrocytes. Neurons were tested next. TX2 again is still shown in red. RBFOX3 positive nuclei, or neurons, are shown in green, and DAPI stained nuclei are shown in blue. And when I enlarge this merged image, what you can see is a clear RBFOX3 positive neuron with TX2 appearing to tightly surround the nucleus, which does suggest that TX2 is cytoplasmically restricted within RBFOX3 positive neurons. Next were oligodendrocytes. And TX2 again is shown in red. Oligo2 positive oligodendrocytes are shown in green. And the merged image, which I will expand right here, shows that in this clear oligodendrocyte, there appears to again be no TX2 co-localization or expression in these cell types. So the main conclusion from this is that TX2 appears to be cytoplasmically restricted within GFAP, within IBA1 positive microglia and RBFOX3 positive neurons. The significant results that presented themselves are shown here. Again, my collaborators produced all of the data and I produced the molecular analysis. I will go over what these results mean. Every other day 2-bottle choice is a measure of the escalation of drinking behavior. And there was a reduced ethanol consumption and preference observed, specifically in males and specifically at 15%. The loss of riding response is a measure of the sedative and hypnotic effects of ethanol and other pharmacological agents. And what we found was that there is reduced sensitivity in our TX2 mutant mice to ethanol, as well as GABAergic agonists, gaboxidol and zolpidem. Chronic loss of riding response is a measure of tolerance. And we found that there was a lack of tolerance development in our TX2 mice when compared to control. The electrophysiological results measured GABA receptor mediated inhibitory postsynaptic currents and demonstrated that the results are consistent with altered GABA transmission and altered GABA receptor composition. The molecular analysis, which again is what I contributed to this project, revealed that wild type TX2 has a differential gene annotation from the NCBI database. Wild type TX2 is expressed similarly in male and female mice and throughout all brain regions analyzed. TX2 appears to be cytoplasmically restricted within neurons and microglia, and mutant TX2 has normal expression of both the TX2 transcript and OSMR. The hypothesis, again, that was being tested was that TX2 contributes to ethanol drinking behavior and responses to ethanol. The overall conclusions from AIM-1 are that the TX2 mutation reduces the sensitivity and tolerance to ethanol in both male and female mice, and that TX2 reduced ethanol consumption and preference in males only. TX2 likely functions through the inhibitory GABAergic system, showing reduced sensitivity to GABAergic agonists and altered GABA release. And there was the molecular characterization of both the wild type and mutant long non-coding RNA. So there was also some limitations, of course, that accompanied this study. The incorrect annotation, novelty of the target, and low endogenous expression of TX2 did provide some challenges. However, I would like to say again that this project was incredibly collaborative across several different labs and universities. And while this has been successful, it took roughly 11 months to generate the first cohort used for behavioral analysis. And this timeline is not sustainable for analyzing the thousands of genes that can be identified from transcriptome data. So our acquisition of this data has really greatly outpaced our ability to functionally screen novel genes of interest. So is there an employable method that can be used to overcome this bottleneck? And that brings me to the technique that I used throughout AIM-2. To address this limitation, I developed a novel technique, but first I will describe the traditional approach, and this is the method that was used to generate our TX2 mutant mice. Embryos are electroporated with a mixture of Cas9 and guide RNA, and the first generation is then wild type, however, they harbor variable mutations. A mouse with an activating mutation of interest is then bred to produce heterozygous offspring, which are then interbred to produce a homozygous generation 3 that are suitable for behavioral phenotyping. Now, this takes a minimum of 9 months and extensive maintenance, and there's still the possibility for the mutation to be inconsequential. So as a means to overcome this limitation, I developed a novel approach to rapidly screen genes of interest to determine the relevance to ethanol action, which I have endearingly named the crispy taco approach. The crispy taco method for creating mutant mouse lines uses multiple guide RNAs and an increased concentration of Cas9 to really mutate these embryos. And using this technique, most F0 mice will harbor a biallelic mutation of interest that will functionally mutate the gene and then are suitable for behavioral phenotyping. So this methodology allows for candidate genes to be quickly screened for changes in phenotypic behavior and then either deemed suitable for generation of a true breeding line and in-depth molecular analysis or removed from further testing. So again, 9 months versus 3 months to get a functional knockout mouse. At the DNA level, guide RNAs are introduced to a genomic region of interest, and these embryos and guide RNAs are then electroporated. So in this situation, let's say we have 3 different embryos, and while each embryo is mutated around the region of interest, each one differs depending on the specific guide RNAs that have been used. These then are implanted into surrogate dams, and once they reach adulthood, they are suitable for behavioral phenotyping. So this technique holds high significance and potential for the genetic engineering field as a whole. And to validate this technique, MYD88, a gene that has been previously characterized to have altered ethanol drinking and ethanol-related behavior, was selected. Exon 3 of MYD88 has been shown to be critical for function. So 4 guide RNAs were designed to be tiled across this region of interest. And when I show the DNA PCR results, the wild type gene is shown, the wild type product is shown on the left, and you can see the variability produced in all of the mutant MYD88 taco mice. And the important takeaway is that while the majority of mutants do not harbor the same mutation, none of them are the same as wild type and are therefore hypothesized to manifest the same behavior. When considering the RNA level, multiple tacos were, MYD88 tacos were used to consider their RNA. And while we see variability in the RNA produced when compared to wild type, again, none of the transcript products are the same as wild type. So the DNA PCR bands from each animal that was used for behavior was also sequenced to determine the precise mutations. And so what you can see here are the individual animal numbers, the guide RNAs used, and the deletions shown in red and insertions are shown in green. And so while again, each animal may harbor a different mutation, they all largely encompass and remove MYD88 exon 3. So I unfortunately do not have the time to discuss all of the behavioral results that were used to validate this method. However, I will give you the main conclusions. So this is comparisons to previous findings. And what the main takeaway is that the results largely recapitulated previous findings as shown in green and are therefore suitable for behavioral phenotyping. So this technique was validated, published, and used throughout AIM-2. So this brings me to my second aim, that mutation of ethanol responsive long non-coding RNAs identified from hippocampal competing endogenous RNA network analyses are sufficient to decrease ethanol drinking and ethanol related behaviors. To identify these novel genes of interest for functional interrogation, male mice were given a priming injection of either ethanol or saline and placed in a vapor chamber for either vaporized ethanol or room air control for 16 hours a day, 4 days a week for 7 weeks. 24 hours after the final ethanol exposure, the hippocampi were dissected and subjected to microarray analysis. This microarray analysis was then used to develop competing endogenous RNA networks from 4 prominent RNA subtypes to identify specifically long non-coding RNAs that are the most interconnected with other dysregulated genes identified in the array. So again, microRNAs, mRNAs, circular RNAs, and long non-coding RNAs were all used to computationally create these competing endogenous RNA networks. And these RNA subtypes are known to both interact and compete with one another, and often for a shared pool of binding partners. So these highly interconnected long non-coding RNAs were ranked. Each gene was annotated but not biologically characterized. These are their original gene symbols, but I will be referring to them as PIT1, PIT2, PIT3, and PIT4. They were all identified as being significantly upregulated following CIEV exposure. So they're all hypothesized to reduce ethanol consumption. So now that the genes have been identified, it is time to analyze their annotation and design guide RNAs. Using PIT1 first as an example, exon 1, exon 2A and 2B are shown in blue. The probe binding site that initially identified this long non-coding RNA from the array is shown in red. The DNA PCR primers are shown in green. The RNA RT-PCR primers are shown in orange, and the guide RNAs that I designed are shown in yellow. And these guide RNAs, 4 were used for PIT1, and they are tiled roughly 50 to 100 base pairs apart from each other. This was done as well for PIT2. However, this time I used 6 guide RNAs, again, tiled roughly 50 to 100 base pairs away from each other. This was done as well for PIT3 with 4 guide RNAs targeting exon 1 and PIT4 targeting exon 1. So these guide RNA locations were selected in order to disrupt expression of the first exon. So this could alter splicing, function, regulation, etc., just from the variability that can be produced from these guide RNAs. When considering their DNA and RNA analysis, showing first the PIT1 DNA PCR, you can see the control wild type on the left and the variability produced by all the guide RNAs and the mutations that have been harbored by the PIT1 tacos. And again, while all of these may differ from one another, none of them are the same as wild type. At the RNA level, what you can observe is that out of the mice used for RNA analysis, none of them still express exon 1 transcript. However, when you look downstream of the mutation, there is now an alternate transcript variant that is present in all mutants that is not present in wild type. Looking at PIT2 next. Oh, someone mute that. Next, looking at PIT2, the wild type band again is shown on the left, and the variability of RNA or DNA mutations that have been harbored are shown for the various PIT2 tacos. Now, unfortunately, these data also highlight the novelty and unannotated nature of these long non-coding RNA genes, as by the fact that I was able to produce variable and inconsistent RT-PCR results, which did result in my inability to analyze the PIT2 at the RNA level. Considering PIT3 next, similar DNA results, we see variability of mutations, however, none are the same as wild type. And when you look at the RNA, the majority of mice are not expressing the transcripts containing exon 1. However, 2 appear to be expressing one of the 2 transcripts that are present in wild type. Downstream, what you can observe is now multiple differential RNA transcript outcomes. Looking at PIT4 next, very similar DNA results, and now at the RNA level, again, the majority do not express the exon 1 transcript. 2 of them do, however, this one is roughly 20 nucleotides smaller, which is indicative of a harbored internal mutation within the boundaries of the guide RNAs, or within the primer sets. And downstream, what you can observe is a maintained PCR transcript that appears to be similar to the wild type band. So overall, the conclusions from this is that at the DNA level, all mice selected for functional interrogation show variable mutations. However, at the RNA level, differential expression patterns can manifest depending on the gene of interest. So as this was a screen for changes in ethanol consumption, 2 different drinking paradigms were used. One is a measure of binge-like drinking behavior, and another is a measure of the escalation of drinking behavior and consumption over time. So to first analyze binge-like drinking behavior, an assay referred to as drinking in the dark was conducted. Mice are given 20% ethanol for 2 hours into their dark cycle, which is their active phase, and the amount of ethanol consumed is recorded. The ethanol bottle is removed, and 24 hours later, the ethanol bottle is reintroduced, this time for 4 hours, and the amount of ethanol consumed is recorded. Blood is collected immediately following removal of the ethanol tube, so blood ethanol concentrations can be measured. So to consider PIT1 and PIT2 first, you can see PIT1s are on the left and PIT2s are on the right, with the PIT1s shown in red and controls shown in blue. And while there is a difference of sex with males on the left and females on the right, there is no difference of genotype. This held true for PIT2 as well, where there is a significant difference of sex, which is expected. However, there was no difference in genotype between our control or PIT2 males and our control and PIT2 females. This held true for our PIT3 and PIT4 males as well when compared to control, and as well for our female PIT3 and PIT4 as compared to control. To consider next their blood ethanol concentrations following this DID procedure. Similar results presented themselves, where you can see between our control and PIT1 males and females, there is no significant difference of genotype. And this held true as well for PIT2 with no significant difference. And unsurprisingly, there was also no difference between our PIT3 and PIT4 males compared to control and our PIT3 and PIT4 females compared to control, which does indicate that these genes are not involved in acute binge-like drinking behavior or altered ethanol metabolism. The next assay that was conducted is referred to as every other day, 2-bottle choice, and this assay has been validated to increase ethanol drinking behavior over time. So the mice are given 2 sipper bottles, one of water and one of ethanol. The amount of ethanol and water are both recorded. 24 hours later, the ethanol bottle is removed and replaced with water. 24 hours later, one of the water bottles is again replaced with ethanol, and the amount consumed over the 24-hour period is recorded. And the bottles are switched to avoid side preference. So I'll show you first the results for pit 1 and pit 2. Now this was done using a ramping paradigm and then ended with 15% ethanol. So I will show you first the male results. So on the top is male ethanol intake and then male ethanol preference. For pit 1s shown in red, pit 2s are shown in green, and our controls are shown in blue. And you can see there is no significant difference in ethanol intake either during the ramping phase or at the extended 15% ethanol. However, there was a significant difference in male ethanol preference, with both pit 1s and pit 2s showing significant differences in preference when compared to control. Considering females next, a very interesting phenotype presented itself with our pit 1 females shown in red, producing a roughly 20% reduction in ethanol consumption, specifically at 15% ethanol, with our pit 2 females having a roughly 10% reduction. There was, however, no significant difference in female ethanol preference. This was done next for pit 3 and pit 4, and this assay was extended to also include 20% ethanol. I'll show you first the male results, and what you can observe rather clearly here is no significant difference in ethanol intake between our pit 3s shown in red, our pit 4s shown in green, and controls shown in blue. And this held true as well for ethanol preference. When considering the females, however, we can see a significant reduction in ethanol preference between our pit 3 females and our pit 4 females when compared to control, mostly at 20% ethanol, with our pit 3s showing roughly, again, 20% reduction of ethanol preference at 20% ethanol. There was also significant differences in female ethanol preference between our pit 3s, pit 4s, and control females, showing reductions in preference, leading to roughly 10% reduction in ethanol preference for our pit 3s and pit 4 females. So to summarize the results from AIM-2, these ethanol assays were run, and the main conclusions out of 4 gene-targeted groups and 8 cohorts, ethanol intake was significantly reduced in our pit 1, pit 2, pit 3, and pit 4 females. Ethanol preference was altered in our pit 1 and pit 2 males, and on our pit 3 and pit 4 females. Now, however, there was also a difference and reduction in total fluid that accompanied this reduction in ethanol intake. However, when non-ethanol tastings were run and compared, there was no significant difference, as well as no change in total fluid when a sweet tastant and bitter tastant were introduced. So the conclusions from AIM-2, again, the hypothesis was that mutation of these ethanol responsive long non-coding RNAs would decrease ethanol drinking and ethanol related behaviors. What I have found was that the crispy taco approach characterized pit 1, pit 2, pit 3, and pit 4 as reducing ethanol consumption in female mice. The approach also characterized pit 1 and pit 2 at altering ethanol preference, specifically in male mice, and pit 3 at pit 4 at reducing ethanol preference in female mice. Moving forward, pit 4 appears to be an ideal candidate for in-depth molecular analysis, as it produced a roughly 20% reduction in consumption and a roughly 20% reduction in preference at a high concentration of ethanol. There are limitations that are similar to the first AIM. The unverified annotations and novelty of the targets did produce some challenges. However, I would like to mention that the taco approach has been validated and published, as well as all of the results that I have just presented to you now. So the next question was, how could I apply CRISPR-Cas9 to enable acute and brain region specific gene perturbation in adulthood, potentially in a gene that has been a little more well characterized? That brings me to my third AIM, that mutation of the Gas5 promoter in the medial prefrontal cortex during adulthood will increase ethanol drinking and ethanol-related behaviors. The long non-coding RNA Gas5 is heavily linked to the pathogenesis of human disease. Gas5 has been shown to be upregulated in the amygdala of humans who suffered from AUD, and viral mediated dysregulation of Gas5 has been in neurons in the murine nucleus accumbens has been shown to regulate cocaine-related behavior. Gas5 can also bind within the glucocorticoid receptor, acting as a decoy, and GR signaling has been heavily linked to AUD. Further, RNA sequencing from the murine prefrontal cortex following chronic intermittent ethanol vapor exposure showed persistent and consistent downregulation of Gas5 at up to a week following withdrawal, which does show a lasting in vivo response within the medial prefrontal cortex to Gas5. This is from the UCSC Genome Browser showing the annotation for Gas5. The main takeaway from this is that there are 5 different Gas5 transcripts and the very close proximity to neighboring gene ZBTB37 with 1 transcript even overlapping. Gas5 has been shown to have multiple promoters. So in order to avoid the possibility of impacting neighboring gene ZBTB37, the experimentally verified Gas5 promoter was targeted. And when I enlarge this here, what you can see is that out of the 5 transcripts, 2 of them are hypothesized to function under the non-coding RNA promoter. And 2 guide RNAs were designed to bind within this region. And then these guide RNAs were then used to generate a Gas5 promoter targeting AAV virus. The AAV12 mainly targets neurons, and this method also limits the possibility of cellular compensation and global effects. 2 guide RNAs were used, as well as the mCherry fluorescent probe to visualize where expression is occurring. These AAV viruses were injected into Cas9 expressing mice via a stereotaxic apparatus. To validate the mutation at the DNA level, this is a chromatogram showing our control LAC-Z guide RNA transfection with the promoter region highlighted in gray and the 2 Gas5 guide RNA binding sites boxed in green. Underlined in red is what is referred to as a TATA box and is found in the core promoter of many genes. So following transfection with the Gas5 guide RNAs, what you can observe are clear signs of mutation immediately following, again, roughly 3 base pairs upstream of the PAM sequence, showing that mutation has been harbored in the Gas5 non-coding RNA promoter. We also wanted to validate this at the RNA level. So this was first done in cell culture, showing roughly 50% knockdown of Gas5 when compared to LAC-Z control in vitro. This was also tested in vivo in AAV injected mice using a microdissection of the mCherry fluorescing region. And what you can observe is a roughly 40% knockdown of Gas5 when compared to our AAV scrambled controls. Histological analysis was also performed to analyze the injection sites and AAV expression in the injected mice. The prelimbic cortex, which is shown as red circles here, was selected for injection, and the prelimbic cortex is associated with alcohol, cocaine, and heroin use disorders, and it has connections to both the basolateral amygdala and the nucleus accumbens. So this region was of interest and could potentially alter those pathways as well. Mice with improperly placed injections, meaning they were too far posterior, are shown in red X's here, and those mice have been properly removed from analysis. Here I'm showing you 2 representative images visualizing the mCherry expression shown in red, showing that the majority of expression is occurring in the PRL. However, there is some spread to other regions of the medial prefrontal cortex. However, this was expected. Drinking in the dark was the first assay that was tested, which again is a measure of binge-like drinking behavior. This was first done in males, showing that there was no significant difference in the amount of ethanol they consumed or their blood ethanol concentrations following their 4-hour period of binge-like drinking behavior. This was also repeated in females. And what you can see is there is no significant difference in the amount of ethanol consumed by the females. However, there is a trend towards a significant increase in the blood ethanol concentrations of these females. So it was of interest to measure ethanol clearance rates in the females to see if there is a potential difference in metabolism. A large dose of ethanol was given to these female mice, and serial blood collections were taken across a 4-hour time period, showing no significant difference between AAV Gas5 and AAV scrambled control females on ethanol clearance. So moving on to every other day 2-bottle choice, which again is the measure of the escalation of drinking behavior over time. I will show you first the male results, and what you can see are the AAV Gas5 in red across the ramping phenotype, 15% ethanol and 20% ethanol for both intake and preference. And unfortunately, this is not significant. However, it does appear to be trending towards significance, and this may have been unveiled with a potential larger N value. So this is not statistically significant, but it may still hold some biological relevance. This was also done for the females, and you can see there is no significant difference in ethanol intake in our female mice across all ethanol concentrations and time. However, there is a significant reduction in female ethanol preference across all time and concentrations. So following these 2 drinking behaviors, a battery of further behaviors were conducted in order to analyze these mice, but unfortunately, I don't have time to discuss them all in detail. However, I will give you the main conclusions. These 9 tests were run. Drinking in the dark and the blood ethanol concentrations are again a measure of acute binge-like drinking behavior. Every other day 2-bottle choice is a measure of the escalation of drinking behavior. Elevated plasmase is a measure of anxiety-like behavior. Acute functional tolerance is a measure of within session tolerance to ethanol. The loss of riding response is a measure to the sedative and hypnotic effects of ethanol. Ethanol clearance was run to measure ethanol metabolism, and as well as non-ethanol tastings to determine if these results are specific to ethanol or if there are alterations in taste perception. Now, the only behavior of significance was found on the voluntary intermittent ethanol drinking paradigm, showing reduced ethanol preference, specifically in females. The rest of the data was negative. The hypothesis that was being tested for AIM-3 was that mutation of the promoter for Gas5 would increase ethanol drinking and ethanol-related behaviors and reducing the possibility of compensation that can occur. There were minor reductions in ethanol preference observed in females only, and unfortunately, there was not much significance. However, that might still hold some biological relevance for Gas5 in the AUD field. Limitations that accompanied this aim was the variability associated in viral mediated genome editing. It is technically challenging to generate large cohorts for behavioral analysis, and the proximity to neighboring gene ZBTB37 did limit my ability to mutate all 5 possible Gas5 transcripts. So overall, the conclusions are that of the 6 ethanol responsive long non-coding RNA genes researched, TX2, PIT1, PIT2, PIT3, PIT4, and Gas5, all 6 demonstrated the ability to alter ethanol drinking and ethanol-related behavior in C57BL6J mice. All 6 long non-coding RNA gene-targeted groups showed sexually dimorphic behavior, which really underscores the need of female inclusion in addiction research. A variety of different CRISPR-Cas9 genome editing techniques can be applied to research different facets of your target gene. And lastly, long non-coding RNAs also hold the potential to be very specific drug targets due to their tight temporal and spatial control and the fact that they can function within discrete networks. Looking forward to the future, continued characterization of ethanol responsive long non-coding RNA genes of interest via the crispy taco approach, as well as now in-depth molecular characterization of the now partially characterized ethanol responsive genes discussed here. And this can be done using a variety of molecular and genomic techniques. CRISPR-Cas9 can also be used to target and modulate gene expression. This can be done preclinically by using therapeutic intervention for excessive ethanol consumption in animal models, as well as clinically by developing techniques that can reliably cross the blood-brain barrier. And the hope is that we want to be able to modulate neuropsychiatric disorders via CRISPR-Cas9 in humans. And with that, I would love to acknowledge my wonderful lab and my mentor, Dr. Greg Hamanix, Carolyn Ferguson, who is our lab manager and instrumental in all of my projects, all of our past and present Hamanix lab members, the Ferris lab and Dr. Ferris for all he has helped with all of my aims, and the Ferris lab, my funding throughout my time here at the University of Pittsburgh, my wonderful collaborators who were instrumental in AIM-1 and the TX2 project, and of course, my dissertation committee and the chair of my committee, Michael Palladino, Greg, Mary Torgroza, Shirley Hill, and Colleen McClung for everything they have done for me over the past 3-4 years. And I would like to acknowledge all of our labs and bar groups and the department. And with that, I will take questions.
Questions for Sonia?
First of all, great project. There were a lot of difficult things that you've done there that worked out wonderfully.
Thank you.
Related to AIM-2, so you know you took your crispy taco. By the way, so you use your crispy taco to look at something that you've already made in a conventional.
Mhm.
And there seem to be some differences mainly related to sex. Now, and I noticed things when we got in three, you had some sex differences as well.
Yes.
So do you think that maybe crispy taco is helping bring out some of these, like, depending on where your sort of your guide RNAs are revealing some sort of sex difference?
Yes.
Their function.
It's very possible. It's possible that that is the case. We have also used the crispy taco approach. Other lab members have used it for other genes, and I'm not sure if they also showed a lot of sex-specific results. However, I would also like to mention that all of the analysis, the RNA sequencing data and microarray analysis that were used to identify these genes were all identified in males. So the fact that we see a lot of female-specific results could indicate potential differential gene expression or differential functions of these genes between males and females. So I think it's important moving forward to consider both sexes for large transcriptome data, as well as for really kind of unveiling the specific nuances of these genes. Thank you.
I want to say
Thank you.
So my question is, what do you think that affects that mutation on
Right. So that is true. Yes, out of the 5 transcripts, I really only targeted 2 of the transcripts. I originally did try to target all 5 using CRISPR inhibition and dCas9. in order to disrupt the bidirectional promoter that targets both ZBTB37 and Gas5 in order to arrest RNA polymerase from extending. It unfortunately was not functional and didn't really work out very well. I have not looked at ZBTB37 expression following mutation of this promoter specifically, but it could be of interest to look at. It's a roughly ZBTB37 starts roughly 300 base pairs upstream of the specific Gas5 promoter. Yeah, yes. So the glucocorticoid receptor is really important for ethanol action. And Rachel Rice is going to be testing that using exon 11 and 12, which are specific binding sites. So I didn't really test that much. However, there are many studies that show that the Gas5 mRNA binding partners have also been implicated in AUD. So I think it would be of interest to look at the specific transcripts that I likely targeted and look at their specific genomic identity to see if maybe there's differences in microRNA binding that could implicate changes. I would also like to maybe repeat this in a larger group or maybe do a whole brain mutation to have Gas5 knocked out throughout the entire brain to see if we can maybe unveil a larger response than what I found here. But again, I mainly target neurons, and Gas5 is expressed in many glial types as well. So that is another limitation.
Additional questions?
Yeah, I have a quick question. Great job, as everybody said. I was curious, like, I think maybe one of them had no transcript that was made for the RNA, whereas, I mean, I know you have many different sorts. And so, is there any way that we've gone back, like, looked at the behavioral responses and seen the presence of a novel transcript versus an is there any kind of, and sometimes they're the same, like,
So what I think it would be really of interest would be to potentially breaking these into sub genotypes and considering that, of course, this was a screen just to see if there's a change. However, now that we know that they do change behavior, it is of interest to go back in, look at the specific mutations, maybe figure out which exact guide RNAs were used to create each specific potential new transcript and to kind of parse that out. It is a potential future direction and something that was of interest. But what I did think was rather interesting was that both PIT1 and PIT2, it seems the majority of the tacos at the RNA level do seem to express the same transcripts. But yes, if we get a larger group and can parse about the standard deviations and standard error, the means are rather small. So I don't know how much of a difference there would be between the actual genotypes, but I would love to sequence that and see what's going on.
Okay, if there are no further questions, let's thank Sonia one more time. Now the real fun will begin. Good job.
do you think two of them just love it?
Small conference room down by. Yep. Big problem with the exchange that we know some people about.
There's no way to make like.
Start making bigger deletions, it becomes less effective. So that's sufficient knockout.
Everyone, I will say there's a class coming in here in 10 minutes to get set up.
I think in the future, we should spend more time characterizing the gene and talk. Pretty cool motor element, you can knock out.
It's gonna take them.
Yeah, do that like in culture.
One time, it's on some quick screen, through a lot of genes. So it's just something about-
And I mean, some behavior challenges, you would hit it.
Yeah.
So,
Yep.
and if you
Yep.
don't, right, you can get some. Behavioral impact.
That's the whole idea, is screen a bunch of genes on something that has a robust phenotype and then start studying it in detail with some more specific methods. Knock out characteristics.
Actually saw them.
How's it going? Good. You figured out all the sort of stuff for the year?
Yes. All the students quizzes. And, so we got. I was, I met you earlier, but. Yes, but I didn't know exactly. Benny regularly back out here. And so,
Yeah. I'm fine. We ended up one of the girls that did it last year.
yeah.
They- if that's what it means.
Yeah. We do have some communication that,
And then I'm.
five of them. The.
Yeah, and I'm happy, yes.
Yeah, so you're in there.
And actually, last year, my last year, was, remember that? Yeah, it's kind of nice, and I'm a little. So, I'm like, so she signed up for something.
Yeah.
I know, I was so anxious.
I mean, when I used to go.
Yeah, well. This,